Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Well, let's go ahead and get started. So I'd like to thank you for joining us here in the room and virtually. I would like to introduce Dr. Timo Hane from the Nature Publishing Group. He's been here on campus for a series of meetings with a colleague and with various groups from around Microsoft, and we wanted to take the occasion to have him here and give us his insights on the implications for science and the introduction of Web 2.0, and a lot of the things that have been happening over the last year or two have been, I'd say, greatly manifest in the work at Nature. They are greatly innovative, breakthrough implementations on the web, and I think we're going to ask Timo to talk a little bit about what has been done by this publishing firm, and also what they are planning to do in the future. So, with no further ado, I will hand things over to Dr. Hane. Thank you.
Thanks very much, Lee. I really appreciate it. So, thanks very much for the invitation to come and speak here. Tony Hay, Lee's colleague, was kind enough to come and give us a talk in our London offices a few months back, so I'm delighted to return the favour, albeit that Tony is really the world expert on what I'm going to talk about, but I'm delighted to come and visit and talk with you about this. So to provide a little bit of context, Nature Publishing Group is a scientific journal publisher. We publish actually just over 70 titles, including Nature, which is our best known title and our flagship title. But my group within Nature doesn't work directly on the journals, mostly. Most of what I'm going to talk about doesn't relate directly to journals, although I'll give you a few examples of things that do. What we really try and do is look at the web as a communication medium and try and think about how we can use that in a creative way to facilitate scientific communication. And that goes well beyond traditional journal publishing. I'll start off by just giving you a kind of overview of the kinds of things my group works on. Actually, and to put it in context, Nature itself comprises about 700 people around the world, so a relatively small company. And my team is about 25 people or so, mostly in London, but some in the US. And this is the sort of range of things that we get involved in, sort of a schematic to help categorize some of the projects we're involved in. And I would there are three main areas I would characterize: databases, these areas around here that I would call social software generally, and audio video, and then we have some sort of supplementary things we get involved in as well that I'll be talking about less. Databases, we have a number of collaborations with academic groups, from the National Cancer Institute to the Allen Institute for Brain Science to UCSD and other institutes, where we work with them on scientific databases. We're trying to apply our traditional editorial and publishing skills to this what to us is a new domain of scientific databases, but is a domain that's as important online, in the online environment, as the journals and the literature itself. So we've got a number of collaborations that we're working on in different areas, and in the examples I give later on, I'll be talking about at least one or two of those. We also have a range of things that we're involved in on what you might call social software. This is really sort of the heart of the Web 2.0 type stuff, participative, sharing, collaborative types of services. And I will be talking about a number of these as we go through the examples that are to come. And we're also increasingly working on audio-video. Had an emphasis on audio over the last couple of years, but we'll be moving forward with video. Again, I'll be talking about that a little bit. We also get involved in some other stuff, looking at web usage statistics, analysing that, and also in some outreach. We hold internal publishing seminars that Tony Hay, that I mentioned earlier on, came and spoke as part of that series. And we organise something called Science Food Camp, which is a collaboration with O'Reilly and Google, and an informal sort of scientific get-together each summer. So that's sort of the range of things we're involved in. What I'm going to do is run through some examples of the projects that we're working on, but also give projects from outside MPG, where projects that particular academics have been working on that interest and inspire us, and I think that point the way towards the way that scientific communication is going to change online. But let's start off by talking about scientific publications, because although I said that my group doesn't concentrate particularly on the journals themselves, they're obviously the core part of our business still, and there's a lot we can do to improve journals in the online environment. So I just want to give a couple of examples and then talk about some sort of more general trends. So one thing is simply linking, and linking the journals to the databases and vice versa. As I said, the online scientific information space, really, I see it's sort of comprised of these two large domains, the literature and the databases. And actually, sort of slightly depressingly, they don't work well together. There's precious little linking between them, and they don't learn enough from one another is one other thing. But just talking about simple example of linking, this is actually one example for us that I think has worked quite well. We have a journal called Nature Chemical Biology where we asked the authors to provide for us structured data about the chemical structures of molecules they talk about in their paper, and then we set up mutual links with PubChem, which is a public chemistry database run by the NCBI, part of NIH. And that actually involves us making a data deposit into PubChem for the structure information. And as a result of that, not only do we create a link into that PubChem record, but they create a link back to us. So anybody looking at that paper can go to PubChem and get more information, not only the information we may have deposited about that molecule, but information that others have deposited there as well. Anyone looking for that information about that molecule in PubChem can also see there's a related Nature Chemical Biology article. It's a very obvious and simple thing to do, but it's very important. Actually, not nearly enough of it goes on. Sort of moving a little bit beyond that, an example from Nature Structural and Molecular Biology. This is a collaboration we have with SDSC, specifically with San Diego Supercomputer Center, where they host this application called First Glance, based on the JMOL software. And this particular development wasn't set up by my team, but was set up by others in MPG. But this allows people reading papers in Nature Structural and Molecular Biology to fire up this Java applet and look at a manipulable 3D rendition of the molecule that they're reading about. And again, this is actually delivered from SDSC, not from the Nature server. So those are sort of simple examples of the kinds of extensions of functionality that we can provide online that we need to be doing more of. But I think they're also sort of what's going on is something that's sort of a bit more subtle and a bit more fundamental, actually, is that as we add more and more links and data and metadata to the articles, what we're seeing is not only better linking between the journals and the databases, but we're seeing the journals become more database-like. So we've already moved from a world in which we've got a print journal to which we've got an online facsimile of that as a PDF. But now we're sort of in the process of moving towards a world in which the text itself, and whether it's literally RDF or SVG as the technologies, the text itself, the figures, actually have some underlying structure. So gene names are labelled and tagged up with unambiguous identifiers, the same for chemical names and species names and things like that, important scientific entities. That the figures are manipulable and the underlying data is available so that you can plot and replot them and download the underlying data for offline analysis. That behind the text we have structured data sets, the data that actually went into producing these results that are described in the text, as well as, of course, metadata to go along with the article. So we're moving sort of from a model in which we've just got a simple online facsimile to a model in which actually the article itself becomes something like a mini database or a data object. And I think also databases can learn from journals, and this is an example actually of a kind of, if you like, a hybrid product that we've created. This is a collaboration we have with UCSD, and a collaboration that started off with a group called the Alliance for Cellular Signaling, the ASCS, who are sort of coming towards the end of their project. That was an NIH Glue Grant funded project that's coming towards the end of its lifespan now. And their goal was to do large-scale, high-throughput biology, gathering information about the interactions of proteins in mammalian cells, the signaling network, if you like, within mammalian cells. And we created a website together with them called the Signaling Gateway. An important part of that website is something we're looking at here called the molecule pages. The molecule pages is a series of database records, between 3,500 and 4,000 of them, about the principal proteins involved in cell signaling, so in signaling within mammalian cells. And you can look at this as in sort of two different ways. It's a database. It's a relational database. It's highly fielded. We'll look at a few more screenshots in a minute. This is actually a screenshot from something that's actually quite textual, but there's lots of other structured data in it, and we'll see some of it in a minute. So it is a database, and it's queryable in the same way a database is. It can be updated in the same way a database is. The information can be viewed in any number of different ways because it is a database. But it's also, for all intents and purposes, really a review publication. It's authored by commissioned expert authors who are experts on the subject of each of these proteins. In this case, G protein alpha S. And you can see, actually, I'm trying to see if the author is listed on this page, but perhaps they're not. But the author is accredited, and we also assign a DOI. So this is a globally unique, stable identifier for this that allows formal citation of this particular publication as well. So it's formally citable. Also, critically, it's peer-reviewed, and that's a large part of Nature's involvement in this project. So all the contents in this database, all the author-entered contents, are peer-reviewed. We send them off virtually. It's all done online. We send them off to anonymous external experts who help us verify the information and correct it before it's published. So, it's citable, it's expert authored, it's peer-reviewed. It's, for all intents and purposes, a review journal. It is a journal, but it also comes in the form of a database. So it's a real hybrid product. And when we started this project, I guess 5 or 6 years ago now, no more than that, probably 6 or 7 years ago, there weren't. It was very, very rare. It still is very rare for databases to be peer-reviewed in any way, for them to be made formally citable and stable in any way. And we're seeing increasingly that is becoming more and more common now. So we're seeing sort of the journals becoming more structured, more database-like, but we're also seeing the databases become more journal-like in some of their editorial approaches. These are a few more screenshots from this same thing. So here we're looking at a particular transition of one state of this protein into another, but you can view it in this sort of tabular form, but you can also view it in a more graphical form at the various different states of the molecule and the transitions between them, and click on these ovals or arrows in order to get information about either the states or the transitions. So it's just showing an example of an underlying structured data set being queryable and the way in which this is really a database as well as being a journal. Another thing that's really key, and that I've mentioned in passing just now, is the process of peer review. So it seems to me that the web as a communication medium potentially allows different modes of peer review, and certainly we've seen some experimentation with that, and we've been doing our own sort of trials with different modes of peer review. So last spring, summer, we launched an open peer review trial for Nature. So the way that this worked is that when you submitted a manuscript to Nature, if it was accepted for peer review, which applies to about a third of the manuscripts we receive for Nature, which actually go through peer review, the others are rejected before review, the author was given the option of us posting their manuscript on a public server and opening it up for comment from anyone who cared to comment. We didn't allow anonymous comments. They were moderated, but otherwise anyone could comment. And over a 4-month period of the trial, the 4-month period of the trial, we had just over 70 papers were published in that way. They were pre-published, if you like, and invited for public comment, which was about somewhere between 5 and 10% of the papers that were submitted over that period, and was an acceptance rate, an uptake rate in line with our expectations. And so here you can see one where we've this is just an example of a comment trail. We posted it was just actually a movable type blog is where we posted it. In effect, though, it was a preprint server, and we invited comments. I don't know whether I've got any stats on this now. We at the end of the 4-month trial, we sort of assessed what had happened. In the end, actually, it turned out that the trial had got a lot of traffic. A lot of people had been reading the manuscripts. A lot of people were aware of the trial. The number of comments and the quality of comments were slightly disappointing, so we decided to stop at that point and not to extend it any further, but we will be doing more experiments of this kind. I think there are a couple of reasons for that, and we can discuss them later if people want to, but I think one of them was that we didn't allow anonymous comments, and scientists are still very sensitive to commenting in public on other people's papers. And the other is that it may well be that just culturally people aren't quite, scientists in particular are actually rather conservative, in my opinion, and they're culturally not quite ready for doing this yet. But I think in time it will become increasingly normal. And we do see other publishers experimenting. Cell Press have been experimenting with public comments on published papers in this instance, not manuscripts that are still under consideration. And other publishers like PLOS, with their PLOS One project, have also enabled commenting on published content. Actually, an important result of our experiment here also was not only looking at the number of comments that came in, but was looking on the impact of those on the editorial decision. And although many of the comments were quite interesting, there weren't any during that period that actually affected our editorial decision about whether or not to publish a paper. And that was also a key sort of success criterion for us in conducting this trial. So it was an interesting trial, and we'll carry on experimenting with those different models, because peer review is of such fundamental importance to what we do as scientific publishers. It's such a fundamental part of the value that we add, that if there are new and better ways of doing it, then we'd better discover them, and we'd better do that first before somebody else does. It's sort of the way I look at it. Here's another. I just think this is a really interesting example of something that's going on outside MPG, outside Nature, that I think is really intriguing and in some ways slightly worrying. This is a really fascinating paper by some sort of, if you like, library science researchers who've written about, certainly at the time of this publication they hadn't actually created, but they've written about a potential algorithm for managing peer review. So the idea here was that people uploading content to an institutional repository, so a database that holds the academic output of an institution, that content is typically not peer-reviewed, but could you in some automated way enable peer reviewing of that content so that it would be scalable? You wouldn't have to have a human running the thing. And so what they've suggested in this paper, in this preprint appropriately enough, is that there could be an algorithm that looks at the authors, looks at the citations, looks at the if you like, the publication, the co-authorship and citation network in that field, and picks appropriate referees automatically based on people who are in that general field but are not so close to the authors that they may be biased, sends off the manuscripts to them, and then asks them to do a quantitative scoring of the manuscript. So this is not a qualitative review of the manuscript, it's a quantitative scoring to help other people decide how important and useful this paper is. That does actually leave out one enormously important part of peer review, which is actually improving the paper, not simply assessing it in some quantitative way as to how good or bad it is. But I just find it an intriguing concept that you would automate that, and naturally enough these kinds of developments and ideas are of great interest to us. I also think that blogs are kind of really interesting in this regard. Scientific blogging has yet to take off in any really big way. There are probably 1,000-odd blogs written by scientists about science, and no more than that. It's certainly, I would venture to say under 2,000, certainly not much over 1,000 at this point. So it's not, in terms of the tens of millions of blogs that are out there, it's not a large number, and it's also a small number in the context of the sort of number of researchers there are in the world, which is somewhere sort of in the low millions. But I think scientific blogging has, well, first of all, just observe that blogging actually has quite a few characteristics that are similar to the literature. Of course, it's much more informal and much more rapid, but people do link or cite, people do build on other people's work, people do critique other people's work. So, the network of blog postings and cross-links are actually analogous to the network of academic papers and citations. Albeit much more immediate and much more informal, but I think for that very reason they have real power. And we are starting to see, albeit in relatively low numbers, scientists blogging and getting real use out of it. But it's yet to become mainstream. And one important reason for that is that scientists don't get credit for blogging. They get credit for publishing in peer-reviewed journals. They don't get credit for blogging or posting comments. And again, I think we might see that change, but that's going to take a while. So we run a bunch of blogs. I mean, so we have. Actually, I don't know how many we have now, but there's certainly sort of around, I would guess, around 20 or so blogs that we run. Some of them associated with our journals. This one in particular is actually just my departmental blog. And we found that some of them work better than others, but generally we found it a really positive experience as a way to engage with people and a way of soliciting feedback and encouraging debate. This is a site called Science Blogs, which is run by Seed Media, another publisher, which I think is doing some particularly interesting things. And they've basically collected together a group of very prominent scientific bloggers, put them on one site, and when you look at the top, an article in Nature recently looked at the top 50 scientific blogs in terms of inbound links, and about half of them were on science blogs. This is a very interesting site. They've got a very good collection of people writing there. All scientists writing about science. So this is an interesting development. This is another really interesting site, which is developed by a guy called Ewan Adey, who at the time he wrote this wasn't working for Nature, but now does work for Nature as part of my team. He created a site called Postgenomiccom. What he wanted to do was to have a way of following the blogs that he was interested in that was a bit more sophisticated than a simple RSS reader. He's a bioinformatician, so hence the site became known as postgenomic.com, but he's now extended the software to cover blogs well outside that domain in physics and chemistry and elsewhere. And what it basically does is it aggregates feeds from several hundred scientific blogs that he sort of hand-picked, and then summarizes which other blogs are they talking about, which papers are they talking about, and gives sort of zeitgeist-type sort of indicators like this. And that's something that we plan to build on in future services that we'll be providing around blogging. So moving away from sort of peer review and blogging and manuscripts, something that's, you know, a new opportunity that's opened up for us as a publisher is obviously in audio and video. So, and that's not only increasing bandwidth, it's also decreasing cost of the software and hardware required to do this. So, you know, we're in a position, as many others are, including individuals, to be not just a publisher, but be something of a broadcaster as well, which is, you know, that's, depending on how you see it, that's either enormously exciting and empowering or completely terrifying. I think there's a mixture of opinions about that among my colleagues, but I think it's very exciting that we have the ability to do audio and video now. Something that we've been pushing very hard over the last sort of couple of years is podcasting, and we've seen enormous immediate uptake in terms of demand for the podcasts. So we started off, we launched a weekly podcast for Nature back in October 2005, and it's come out every week since then. It gets around sort of 40,000 or so downloads a week. We very quickly picked up a sponsor, and so it became self-financing, but also a really important reader service, a really important promotional tool for the journal, and an author service, because what it mainly features are scientists who published in that week's Nature talking about their work. So for us, it's another way of enabling scientists to communicate with other scientists, and it's qualitatively different from reading their work or reading about their work, because you get it's much more informal medium, and you get much more of an idea about the personality behind the science as well. So we found it enormously popular. The other thing is it helps people with is being more productive, because of the audio medium, people can listen to it while they're doing other things, and we can engage with them while they're doing experiments, while they're commuting, while they're working out, whatever it might be. And they can use that time effectively to keep up to date with what we've been publishing. So since then we've, I don't have a screenshot of this, but since then we've launched a whole series of other podcasts, some of which are one-off, others of which are in the process of becoming regular shows, and we've got a dozen or so shows now running. Not weekly like the Nature one, less frequent, or in some cases one-off, but we've also really ramped up our ability to be able to do audio output. We have an audio studio in our London offices. We've trained our journalists and editors in how to do audio. So for us, this has been a real qualitative change in the type of information output that we do. And when you look further afield at the sort of interesting things going on in audio and video, we're certainly interested in video, and I think one of the most promising areas for video in science is actually in methods and protocols, right? So this is, instead of reading a series of step-by-step instructions, or merely reading a set of step-by-step instructions about how to do a particular procedure, you can watch someone do it. This is sort of cookery TV for scientists. And this is a really promising area for video. So, and these are videos, you know, it's not going to be, of course we want to do videos in terms of some of the visually exciting papers that are published in Nature, but we also want to be doing, you know, this kind of thing that enables scientists to understand one another in terms of techniques and methods. And this is a site that we're not involved in, but I think is particularly interesting, the Journal of Visual Experiments, which is a kind of moderated YouTube for scientists and scientific methods. I want to come on and talk about social software now and collaboration. So the web is fundamentally a collaborative medium, and that's really where I think the biggest opportunities of all lie. So I'll talk about some of the projects we're working on, and also some interesting things I think that are going on elsewhere. So we were talking about protocols, methods and protocols. We actually have a journal called Nature Protocols, And that operates in a traditional way that we will commission, or there'll be protocols submitted and they'll be peer reviewed and published. And it's principally an online publication, although there is a print version of it as well. But the thing I really want to talk about here is there's a section of the site that's called Nature Protocols Network, where people can actually upload their own protocols and share them in a sort of YouTube or Wikipedia kind of way, which is lightly moderated. It's not peer reviewed. It allows for much more immediate exchange of information. And something sort of even more radical that's again not associated with nature in this case is something called OpenWet, where I think this is one of the most interesting scientific sites out there. This was developed by a team at MIT working under Drew Endy, a synthetic biologist, and they. Technically what they did was really simple, but socially what they did is really important. So they've just taken a copy of MediaWiki, which is the wiki software that runs Wikipedia, and they use it as their online lab notebook, basically. So they post details of experiments they're running, results that they got, but also things about their sort of diaries, which meetings they're attending, and so on and so forth. There's a video here when I took this screenshot a while back of John Wilbanks from Science Commons giving them a talk, and they've clearly posted a video of that. So this is kind of being radically open. This is really different from what scientists are used to doing. So this is being open to the degree of not only having a website that details all of the things they're doing, including what experiments they're running at the moment and which ones worked and which ones didn't, but actually enables anyone, in principle at least, to edit that information. And they have, since then the site has grown, and they've got several hundred users from various labs around the world. I think talking to them, they also find one of the challenges is they built this thing because they wanted to use it, and now they found that it's actually quite popular, and they're wondering, they want to concentrate on the research, they want to concentrate on running this site. So that's one of the challenges they've come across. But I think it's been a really interesting and in its own terms quite successful experiment in sort of radically opening up science. But it's still a minority activity, this kind of thing. Scientists, particularly biologists, tend to be rather proprietary and often very secretive about what they're working on at any particular time. Talking of relatively proprietary areas of science, chemistry is quite a proprietary area of science, and this is an example of someone trying to do something rather similar in chemistry. This is Jean-Claude Bradley's group at Drexel University, who have something very similar. They have a wiki-based online laboratory notebook about all the work they're doing. So again, they're being radically open about what they're doing. Moving on to some of our projects again, this is a screenshot from something called CanaTier, which is a social bookmarking service we'd set up for scientists. So for those of you familiar with Delicious, it's inspired directly by Delicious. But when we saw Delicious first time many years ago, we thought it was a fantastic idea, but we also thought there were certain bits of functionality that could be added to that software in order to make it more relevant and useful to scientists and other academics. So for example, when you bookmark, so the way that it works for people who aren't familiar with Delicious is when you're browsing around the web, you install a little bookmarklet, which would appear, here it is on my browser, and when you're browsing around the web, you just click on that, a little pop-up will come up, and you can enter some tags into it, and then without having to actually come to the CanaTier site, it will save that in your account, and you can come back to your account later on. And in this case, I'm looking at the things I've bookmarked under the tag called systems biology. But what we've done is to build in some functionality that, for example, when you bookmark a link on a known academic site, so it might be something like PubMed Abstract database or Nature.com or another sort of well-known academic site, it will automatically import the metadata associated with it. So it will remember who the authors are, what the journal is, and when it was published, and so on and so forth, things like the PubMed identifier. So it will remember important bits of information so that you can retrieve that then later based on that. And we code that to work sort of individually with each site. So each site we kind of know how to retrieve that data, and CanaTier does that automatically on behalf of the user. There's a whole bunch of other functionality we've built in that's specific for scientific users, but that's a kind of example of the sort of thing we've done. And CanaTier is not only a useful way to store bookmarks for your own purpose, but you can share them with one another. So if people want to see what I've bookmarked under systems biology, or to give a better example in the sense of something that people do actually use quite widely, we have a journalist called Declan Butler who writes a lot about avian flu, and he keeps a kind of a list of stuff that he's reading on avian flu, and people follow what he's posting there, and you can follow it because each page has an associated RSS feed, so every page has an RSS feed. We've also created an API, and so that other people can build things on top of it. And they have done. This is actually an example of something, a collaboration we did with the developers of ePrints. So ePrints is an institutional repository software developed at the University of Southampton and very widely used. And this is actually a screenshot from their instance of it at the computer lab at the University of Southampton. And this is, so the screen is from a particular manuscript, and if we were to look up, there'd be the abstract and a link to the actual PDF or whatever it is that's been uploaded. And what we're looking at is the bottom of the screen, which is an extension we created for ePrints that provides CanaTier functionality. So this is trying to use the tags in CanaTier and the functionality of CanaTier to provide some infrastructure and functionality for third-party sites. So what do you get from here? You get a list of tags that people have used in CanaTier to describe this particular item. Okay? If you were to click on those, it would then expand to show you other items in CanaTier that have been tagged with that same term. So that's a way of pulling further information out of the system on similar topics to the paper you're currently reading. If I were to click on that little link there, it would pop up a little form, a sort of AJAX type interface, pop up a little form that would allow me within the context of this particular site to tag and save this item, this URL, into my CanaTier account. And then further down here we have a more generic kind of related reading list, which is based on not any specific tag as these ones would be based on, but a general coincidence of tags and users of people who've bookmarked this. So on that basis, a kind of collaborative filtering type algorithm, rather like you might see on Amazon and other sites, saying based on people's global bookmarking behaviour, what other stuff is in CanaTier that's similar to this. And so we're able, sort of through that sort of wisdom of crowds type approach, to provide a new piece of functionality for this site that doesn't exist before. One of the problems with institutional repositories, at least in my opinion, is that the individual articles are kind of very standalone, and it's very difficult to go to an article and find other similar stuff, because typically other similar stuff will be elsewhere around the web, not just in that repository. So it's good for retrieving specific articles, but it's not good for browsing around and serendipity, and we're trying to provide that kind of functionality here, using the data that's in CanaTier, and at the same time trying to make CanaTier into something of a piece of infrastructure that provides functionality for other sites, not just for CanaTier itself. Much more explicitly, when you talk about social software, a lot of people obviously think about social networking. We do have something we've launched in its full form actually just a few weeks ago called Nature Network. And Nature Network is it's a number of things actually, but it is a social networking application for scientists, so it's sort of like a sort of a LinkedIn for scientists, if you like. But it also goes much further than that. It's you can create groups and discussions, and in fact the principal reason for creating a network of people within the system is so that you can follow their contributions to the various discussions that are going on around the site. It's also a blogging platform where we have invited bloggers who write on various topics, and it has two portals, actually, two city-specific portals. We've started off with Boston and London. You see a screenshot from Boston here, which not only has that sort of networking functionality, but also has local news, blogs, jobs information, events information as portals into the system. And what we're trying to do here is create, it's really very much aligned to the Web 2.0 concept, the architecture of participation concept, building up network effects. So this and CanaTier and other things that we're working on are really trying to create sites that get better for everyone the more people use them. And that sort of seems to me to be key to making the most of the web, which of course is a network itself. And so building network effects is a particularly effective way of adding value. I also want to talk about open data. So open data, I mentioned earlier on the CanaTier API, allowing people to build on top of that data and functionality. But we can also do similar things with our content. And so one very simple way, we're among the earlier adopters in scientific publishing of RSS, and we think RSS, it immediately struck us as something very useful and powerful. As we were discussing with Lee and his colleagues earlier today, it's still too hard for people to use. The applications that use it still aren't quite refined enough for sort of really truly mainstream use. But RSS itself is a wonderful idea. But what we did when we issued our RSS feeds, and we have RSS feeds of all of our journals and lots more besides, is we tried to include in them, and sorry, let me just backtrack. So of course those RSS feeds can be used in a standard RSS reader. Here's a screenshot from one that I used to use. But when you look inside our feeds, we're actually including a lot more data than you'd actually find in an ordinary RSS feed. So an ordinary RSS feed, you would get a title, a link, and a description. So in this case, it's an abstract. But we're also including a lot more structured data about, this is doubling core metadata, about the title and authors and so on and so forth. And also, Prism is a metadata format that came out, metadata standard that came out of the magazine publishing industry. And so we've included this doubling core and Prism metadata. And the great thing about it is that the standard RSS feed readers will just ignore that. So it's sort of wasted bandwidth, but it doesn't really do anyone any harm. But we've put that data in there and people can use it. And what we saw happening then was people, particularly library information systems, making use of that metadata, and other publishers adopting this form of extension to their RSS feeds and including the same kind of information, which enabled these library information systems that were really interested in this kind of data to ingest that as well. So our RSS feeds were serving the sort of dual purpose of just general purpose RSS feeds for people with ordinary desktop readers, but readers with more sophisticated information needs and interests could also ingest this highly structured information that was really specific to our domain. And we sort of took that a step further with something called OPMI, or Open Text Mining Interface. So this was an idea that people regularly contact Nature asking for, so these are researchers asking for our content, saying, could we text mine it, please? And of course also, search engines, of course, like, including Microsoft's own search engines come and index the content. And that's sort of fine at one level in that we can, with Microsoft and with Google and Yahoo, we can enter into sort of bilateral agreements in which, we let them in to index our content. Once you start moving to researchers and others, it becomes really difficult to scale. I mean, at one level, it's a pain for us to have to serve lots and lots of different requests for the content, which we usually try and meet as far as we can. But even more for the researchers concerned, for them, it's a real pain to get a big body of content from lots of different publishers. They have to approach all these different publishers and try and get hold of this content. So, of course, one solution to that problem is just to make all the content freely and openly available, and the open access movement, or parts of the open access movement, have been pushing towards that. But I think, for the foreseeable future, realistically, we're going to be in a mixed economy where some content is going to be reader pays and some content is going to be author pays and available for mining and reading freely. So we thought, well, what can we do in the here and now to really enable people to index our content, to mine our content, without having to contact us and ask us for permission or ask us to send them the XML. And it struck us that there are actually some bits of information or some types of information we could just open out that would be useful for some types of indexing, but wouldn't jeopardize the subscription business model, because they wouldn't be particularly useful for people for humans to read. So OPMI was our sort of proposal, and it's still really at the proposal stage. We have it implemented for our content. We have some research groups working and building demos on it, and we have some other publishers interested, but it's hardly an industry standard at this point. And we could provide a few different things. We provide basic metadata of the kind I showed you in the RSS feed, and this is off the top of this particular screenshot. We also very simply provide a list of word vectors. So we provide a list of the words that appear in that document and the number of occurrences, and there's also a stop list of very common words. And that's useful for very, very basic types of indexing. You could build a full text index off the back of that. You can also do slightly more subtle things like latent semantic indexing to do document similarity clustering. But beyond that, of course, lots of indexing and text mining goes well beyond individual words. But a lot of it, particularly a lot of it used in academia, doesn't go beyond sentence boundaries very often. So another thing that we did was we put in these OPMI files what we called snippets, which approximately correspond to sentences. So these are basically sentences, all the sentences in the article, but they're out of order. So they're not particularly useful for someone to read. But if you're doing text mining using algorithms that stops at sentence boundaries, it's every bit as good really as having the full human readable text. And again, we can just put that out there for people to use as they see fit. I'll skip that one actually. Another. Let me go back and cover that one. So that's OPMI, and that's sort of starting to progress, and we've got good interest from a number of research groups in that. This is sort of an example from chemistry, where there are particular problems with chemical data, and one of the problems actually is that the principal naming system used in chemistry, the CAS numbers, owned by the American Chemical Society, proprietary. They are owned by the American Chemical Society. So one of the things that's going on in chemistry at the moment is there's a movement to use sort of alternative identifiers, and one of the main ones that's gaining some support are called INCHIs, International Chemical Identifiers, I think it stands for. And there are a couple of benefits to INCHIs. They're non-proprietary, but they're also descriptive of the structure. They're not just arbitrary, opaque numbers. And this is actually a demo out of Peter Murray-Rust's team in Cambridge, UK, where you can use this application, this little applet here, to build a chemical structure. And it will then work out the INCHI identifier for that chemical structure. It's just computationally derived from the structure itself, and then conduct a Google search for that INCHI. And because INCHIs are, I mean, if you see an INCHI, it's usually quite a long string, and there's no way you're ever going to find that string used in any other context than chemistry. And for that particular molecule, it's a very reliable indicator of information about that molecule. So this is basically a kind of a structure-based search engine. It's a very simple piece of software, working off the back of Google, but using this INCHI identifying system in order to identify information about particular molecules. I think it's a particularly neat example of something that's very simple but potentially very powerful. And this is, I think, one of my favourite examples of sort of open data. And this was done by Declan Butler, who's the journalist who works at Nature, who I actually mentioned a little bit earlier. And as I said, he covers a number of different topics, but one of the things he works on is avian flu. And he noticed when he was covering avian flu that the data about outbreaks is very fragmented and often secret. So there are many bodies that gather information about avian flu outbreaks that actually promise to governments that provide it to them not to make it public, because governments tend to be worried about things like tourist industries when they report avian flu outbreaks. And so, but even more than that, even more than than much of the data being secret, it was very fragmented. It was in lots of different places, in lots of different formats, and there was no really useful way of visualizing that information and interpreting it. So what Declan did, and bear in mind he's not a researcher, he's a journalist, albeit a kind of a hyperactive and particularly sort of technologically savvy journalist, but he sort of felt that this was wrong and he wanted to do something to sort of fix that. So he gathered all the avian flu outbreak data he could find, and he put it in a KML file. So KML, of course, is the XML standard that Google Earth uses, and made it visible in Google Earth. So at a stroke now, the information, so he just posted it on his blog, the information is available in a structured, usable form, all in one place, and you can look at it through this wonderful interface. And immediately you can interpret this information much more effectively than you ever could, even if it had been in one place before, which it hadn't been. He then went further, and he obtained poultry density data, which he overlaid on that. So now you can look at the outbreak information and look at the poultry densities as well. So red is high, yellow is medium, these grey areas are low. And immediately, I mean, even me looking at it as a non-expert, you can see it makes sense, right? I mean, you know, the data makes sense in a much more immediate and profound way than they ever did when it was a list of numbers or held in different web servers around the world. You know, this data set has become a really kind of important research resource now. I think it's a great example of someone with a really good idea, who's motivated and clueful, who can have a massive impact with you know, a relatively small amount of effort. So I think that's one of my favourite examples of open data. I think it's a particularly neat example. I want to wrap up by talking about virtual worlds and specifically about Second Life. This is an area that we're kind of very much engaged in. It's really at the, you know, I started off talking about journals, which is our mainstream business, and went through social software, which is very much an evolution for us, and something of an experiment. And this is really the kind of the real far experimental end of what we're working on. But I think all the more interesting probably for that very reason. And so we, I think Second Life and other virtual worlds like Second Life hold enormous potential for scientific communication, visualization, education. We don't yet know exactly how it ought to be used, but until we start experimenting, we'll never know. So that's the sort of approach we're taking. So we got some land, we got ourselves an island. Inevitably, we called it Second Nature, and we decided we wanted to give it a scientifically meaningful shape without making it too hard to terraform it. So we gave it the shape of a water molecule. So we have the oxygen and the two hydrogens, and to start off with, it was empty. And then we thought about what application could we create. And when you think about 3D representation of scientific information, probably the simplest type of stuff to do are molecules. So we created something called the magical molecular model maker, or M4, and we made it look like a bubblegum machine, but actually what it does when you make a request is you give it a molecule name or identifier, it goes off over the web to PubChem, the NIH database of chemical structures, and then it creates you a 3D representation of that molecule. And the thinking is that this kind of thing, if you were to run a seminar in Second Life, or some kind of meeting or discussion, and if you were talking about chemical entities, chemicals and chemical structures, you know, it would be really useful as you talked about this to be able to pull up any chemical structure at will while you're talking about it and have other people be able to look at what you're talking about. This is something that people would love to have in the real world, but we don't, unfortunately, and it's very hard to create in the real world. You know, in Second Life, this was a very few days' work for an able developer. So that's sort of where we started. These are some screenshots I actually took, I think, yesterday of our first island. So you can see there's one of the hydrogens. This is the main oxygen bit to the island. And we started now to get a whole bunch of activity. So this is most of this, almost all of this, in fact, has been created by other people, not by us. We started off creating this chemical thing. We created a few other things as well. We created some an application based on the Sloan Digital Sky Survey, which was kind of a virtual telescope that you could point in different directions and it would pull the appropriate image off the Sloan Digital Sky Survey website, and a couple of other things. But since then, we've sort of attracted a bunch of scientists who are active in Second Life, and what we're trying to do is provide an environment in which they can experiment with things. If they're doing something that's scientifically relevant and we think it's interesting, we're happy to give them space on the island to try it out. So there's a human chromosome display here. This, I actually forget exactly what, this is another genetics display here. This is a virtual cell. This is actually Jean-Claude Bradley, who I mentioned earlier on, who runs the Useful Chem lab notebook wiki at Drexel. His team created this. So this is a sort of chemistry-oriented building, and actually he's using this. This is his area as well, which is these blue obelisks are actually multi-choice kind of. It's actually a virtual multi-choice questionnaire that he uses with his students. And I'm trying to see what else we can see. There's a meeting area over here. I've got a few more shots to see some of the other things. That's the chromosome, human chromosome display there that we saw earlier. Off in the background, you can actually see a second island that we got. We started running out of space, and we've got another bunch of people, earth scientists and chemists, working on this second island here, doing some various demos. And I'm not even 100% sure exactly what they're building at the moment, but I'd have to check with my colleagues as to where they're at with that. But I think there's some earth science stuff going on, things like glacier demos and so forth. Here's an area where you can pick up molecular structures and find out information about molecules. Here's an area where you can get an RSS feed of news in from the Nature site. This is an area created by Yoey May, who's a scientist who created a way of doing a poster session in World, right? And this is actually, I can't remember quite how far she's got with the development, but the way it either already works or is intended to work is more clever than simply you paste an image onto this poster. It actually goes off and pulls the information from web pages and formats it appropriately on the poster display. And she also created a PowerPoint viewer, so you can give presentations of the kind I'm giving now. And then this was another thing that we experimented with. So this is a figure from a paper in a journal we published called Nature Reviews Molecular Cell Biology. It was done by a group at the European Molecular Biology Lab, and what it is, it's an electron tomograph of a bacterium. So it's a computer reconstructed view of a bacterium composed of slices, many slices that were made through the bacterium and then scanned with an electron microscope and then reconstructed in this sort of pseudo 3D form. And this seemed to us to be a particularly interesting type of figure to use in a virtual environment. So we thought it would be great to be able to create this, but in such a way that you could actually step in there and wander around and point to things and discuss things with people rather than have this flat pseudo 3D image. And luckily, the group who were doing it, Rob Russell and his team at EMBL, were also really interested in ways of using Second Life for scientific communication. So they gave us the data, and we worked on this. So the rendition here, as you can see, is much lower resolution than you see in the journal figure. That's because of limits on the number of prims or individual shapes that you can have within on a certain island, on a specific island in Second Life. So the technology is still limiting us. But as as a sort of test of principle, I think it's an interesting and successful experiment in that you can actually, even at this low resolution, get much more of a feel of the 3D layout of this bacterium than you can from the flat pseudo 3D image. And actually, interestingly, the renderer that we created, or that a developer called Matt Bidoff created for us in order to create this cell here, is actually a generic renderer. So we were able to get other data sets, and this is an ideal knot. This is a mathematical structure now, but is relevant to scientists in various domains, from quantum physics to biochemistry. And it again seems to me to be a particularly interesting example of something where this kind of visualization tool is just a much more powerful way of conveying the concept than any other means that you might choose to think of. So there's lots of other scientific stuff going on in Second Life that's not going on our island. I mean, there's the Spaceflight Museum, I think, is one of the most amazing places in Second Life, where you get replicas of all the manned spaceflight vehicles, and you can climb into one of the rockets and go up into orbit and see a demo or the display of the space shuttle. There's the four-dimensional house, which I think is another fantastic example of being able to convey very complicated concepts quite simply in Second Life. So this is a house that, as you walk through it, rearranges itself to give the impression that it actually has 4 dimensions. So as you walk straight through the house, you end up coming back to rooms that you had previously visited, even without turning around. And another one of my favourites is the schizophrenia house, which was created to teach particularly carers and families what it's like to experience schizophrenia. So this is a house where you can go in and explore it and it's a sort of ordinary, innocuous-looking house. You can go in and explore it as a healthy person, but then you can flip a switch and experience what it's like to be schizophrenic, and you sort of see psychedelic patterns and hear voices and things that, words or paintings on the walls that were previously innocuous come over as threatening and aggressive. And again, it's a sort of immersive experience that gives people a much more profound understanding of what it's like to have that disorder than you could achieve in sort of other media. So I think that Second Life and worlds like it, albeit they're going through, and Second Life in particular is going through various growing pains at the moment, hold out a lot of promise for scientific communication. I could easily imagine it being used as virtual conference setting, virtual lecture settings, enabling people who are separated geographically to come together, but also using these kinds of visualization tools in order to be able to convey things more effectively than you would otherwise be able to do. Not only than you'd be able to do on, say, a telephone conference, but possibly in some cases more effectively than you'd be able to do giving a lecture like this in real life, because the possibilities are so endless when in terms of creating these kinds of visualizations. So I just want to wrap up by talking about some of the big trends, that are kind of implied by what I've been talking about. We are going, there's sort of, if you like, a shift from journals to databases in terms of the relative importance. In the past, databases, if you go back a couple of decades, databases were really relatively unimportant to most researchers. They're becoming more and more important in a variety of domains. And in some domains and to some researchers, there's no question that databases are more important to their work than the journals are at the moment. But I think more subtly, as I hope I conveyed earlier on, journals and databases are also becoming more like one another. We're going, and in that sort of context, we're going from static text to interactive data. This is a really important and profound shift for us as publishers. Science itself is going from cottage industry to industrial scale, and that's particularly in biology over the last few years. We see big projects of the kind that some of them the kind that we've been collaborating with, people like the Alliance for Cellular Signaling, who I mentioned earlier, people like the Allen Institute for Brain Science in Seattle, another group with whom we're collaborating, who are doing these enormous industrial scale experiments and churning out data. So you're suddenly seeing a degree of specialization in which there's whole enormous teams, almost factories, churning out data, and other people then doing analyses and building insights on the basis of that data. So you're seeing greater specialization and a kind of an industrialization of science, industrialization in particular of biology, as opposed to this cottage industry model where individual labs are doing everything from gathering the data to publishing the paper. And of course, the web being the kind of environment it is, we're going from a closed world into an open world. And I've given you some examples of some of the scientists who are opening up and kind of exposing the internal workings of their labs and exactly what they're up to. And it's certainly true to say that that's not generally the case, but it's becoming more and more the case. And it seems to me that it will continue to progress in that direction, not least because that's good for science and the discovery process itself. And with that, I'll stop, and I'd be happy to take any questions if there are any. Thanks. Lee.
You've touched on it, I think, in a
variety of different ways, but one thing that I've been very interested in is the release of things like Swivel, Freebase, etc. I'm wondering about what the impact is on peer review, on open access, quality of data, etc., related to those new tools.
Yeah, I mean, well, I think the impact, we don't know yet, of course, because they're very recently released. I think, clearly those kinds of tools, whether those, what happens to those specific examples, we'll have to wait and see. But those kinds of tools have got to be useful in I think, even more than making the data available, in helping people understand it and collaboratively analyse it. I think I haven't actually spent a great deal of time on Swivel, but it looks to me that things like Swivel and Many Eyes are kind of really interesting developments in that area. And it seems to me sort of self-evident that those kinds of tools will help people collaboratively, share and analyse the data. So I think those are important developments. We'll have to see what happens to those specific projects. Freebase is something I've looked at a little bit more, and it's something a bit different. But it's kind of it's a sort of a structured Wikipedia, if you like. So where that people can put in their own information, but that holds sort of instead of just holding HTML information, holds sort of unambiguous semantic relationships between the entities in the database. I find that an absolutely fascinating project, and we've been talking with them about, what is it we could do in terms of getting some scientific information in there. But I think even that team, the MetaWeb team who created it, are still trying to get their heads around exactly how to do that. So I think it's too early to say with Freebase exactly what's going to happen there in the scientific context. Again, a really interesting experiment that's going to, or a really interesting project that's going to potentially change things around, but I think it's too early to say. Okay, thank you very much.
Thank you very much.